1 billion people have parasitic worms living inside of them. That is billion with a B, making it around 1 in 8 people worldwide. Hello, I'm Dr. Astra Bryant. I'm a research fellow in the lab of Alyssa Hallam at UCLA, where I study how parasitic worms hunt for human and animal hosts by sensing our body heat and odor. The worms that I study are a major source of neglected tropical disease. They're spread by contaminated soil and water and include species such as hookworms and the human threadworm. Our ability to prevent infections is extremely limited and in pretty desperate need of innovation. My research seeks to enable the development of new therapies by understanding how parasitic worms use their sense of smell and their ability to sense heat sources to find and infect hosts. Worms such as hookworms and threadworms are soil-transmitted parasites, which means that they're infective as small larvae living outside of a host. Their goal is to navigate through the environment until they find a suitable host. Once a host is located, the larvae infect, often by penetrating the skin of the feet. How do these larvae, which are less than 1 millimeter long, find hosts? We know from researchers studying mosquitoes that body heat and odor can be powerful cues signaling the presence of a warm-blooded animal. Do parasitic worms use these same senses, heat and smell, to guide their host-seeking behavior? So to test whether parasitic worms are attracted to heat, I built a heated arena that allows us to record the response of infective larvae to precisely controlled thermal gradients. Together, my team and I are using the setup to test the temperature-driven movement of a number of parasitic worm species. We've found that the infective larvae of parasitic worms move really rapidly towards heat sources. And to give you a sense of what this looks like, I brought a video. So what you'll see are the infective larvae of the human threadworm moving in a linear thermal gradient. Each worm is 0.5 millimeter long. Despite their diminutive size, in under 10 minutes, these worms can travel almost 300 times their body length in search of warmth. This is the most robust sensory-driven behavior the Hallam Lab has ever observed in any worm species. Human threadworms are pretty exceptional heat seekers. So we've repeated this experiment in a number of different parasitic worm species. We find that in general, the infective larvae of mammalian parasitic worms prefer to be at or a little bit above human body temperature. So all our experiments are confirming our hypothesis that heat acts as a potent host cue for parasitic worms. Now, in addition to their thermophilic or heat-loving tendencies, many parasitic worm species are also attracted to the smell of a mammalian body. Are these scents interchangeable with thermal cues? Or are these two senses ranked in a hierarchy, as they are for mosquitoes? To answer this question, we're testing the effect of heat on the response of infective larvae to known host odors. We've found that the infective larvae of the human threadworm that you just saw will ignore an attractive smell in favor of moving towards warmth. However, when we heat that same smell to the temperature of a human body, the worms now migrate towards us. This tells us that yes, parasitic worms have a hierarchy of host cues. Furthermore, it helps us build an order of operations for how the infective larvae find hosts in their environment. Our updated model is that infective larvae first use their sense of heat to detect the presence of a potential host in their local environment. Moving towards warmth, they then use their sense of smell to determine whether that heat source is a member of their preferred host species. Interestingly, this hierarchy is the reverse of what we see in mosquitoes. By understanding what sensory cues are necessary for parasitic worms to find their hosts, we're now in a better position to design effective worm traps and develop public health recommendations for preventing infections. So what's next for our research? We have many questions. As a neuroscientist by training, I'm interested in how the brains of these parasitic worms produce such dramatic host-seeking behaviors. Are there sensory circuits specialized for detecting host-emitted sensory cues? How are the brains and even genes of these parasitic worms different from those of free-living worms that avoid warmth? What are the cellular and molecular adaptations that allow parasitic worms to respond so dramatically to the presence of a host animal? By understanding the neural basis of how parasitic worms use sensory cues to find hosts, can we identify new ways to prevent infections? We hope the answer to this final question is yes. As we learn more about the neural basis of parasitism in these worms, we in the Hallam Lab are optimistic that our research will ultimately provide critical insight, leading to the development of new therapeutic strategies for treating or preventing one of the world's most neglected sources of chronic disease.